Which among the following method is used for the bioassay of auxin hormone? The auxin hormone can be bioassayed by observing the curvature of the Avena coleoptile, which is the young shoot of a grass seedling such as oats, and this method is known as the Avena coleoptile curvature assay, and this based on the response of coleoptile to auxin hormone. So initially we have to cut off the Avena coleoptile and place it on a agar block, and then the auxin diffuses into the agar block, and we will place the agar block asymmetrically on the cut coleoptile stump. And later we can see that the coleoptile is showing typical curvature. That is, when auxin is applied to one side of the coleoptile, it causes that side to elongate more than the opposite side, resulting in curvature. So that means the correct answer is option B, Avena coleoptile curvature.